An introduction to bioinstrumentation. In the last lecture, we talked about the basic bioinstrumentation system and the baseline for most bioinstruments. In this lecture, we'll be talking about the parameters that these bioinstruments collect, as well as sensors, in particular electrodes. Before we start, let's talk about the parameters and the variables that these bioinstruments collect in order to understand the patient's health better. These include biopotentials, pressures, flows such as blood and air, displacement, impedance, temperature, as well as chemical concentration of various chemicals and molecules within the body. There are two main types of measurements in order to get these parameters: in vitro and in vivo. In vitro is defined as out-of-body experiments, while in vivo is in-body experiments. For in vivo, the sensor packaging is very important because it needs to be safe and fully functional and reliable. In addition, the biocompatibility, or how well the instrument works with the rest of the body, as well as the materials of the instrument, are very important because it needs a long operational lifetime, as it is very difficult to do the surgeries and intrusions needed to get the instrument in and out of the body safely. To get into the biopotential sensors, we'll talk a little bit about electrodes. Electrodes are used to transform ionic potentials generated inside the body to electrical currents. These electrical currents can then be measured and used to generate experimental results. Electrodes must record accurate, stable readings that are undistorted to provide correct data. The metal of the electrode specifically is very important, as it needs to be corrosion resistive and not damage the patient. Implantable electrodes have relatively strong metal electrodes in order not to break when they're inside the body. There are two main types of electrodes that we'll discuss. Non-invasive electrodes go outside the body, while invasive electrodes go inside the body, specifically on cells and other tissues. Let's talk a little bit about how ionic pulses are generated into electrical signals. The electrode serves as a transducer to change an ionic into an electric current. The junction between the electrode and the electrolyte allows for the transduction to take place due to either reduction or oxidation reactions. The flows of ions in the electrolyte gives rise to flow of electrons in the electrode due to a reaction at the interface, as mentioned before, either an oxidation or reduction reaction, depending on the direction of the current flow that occurs at the interface. From there, the electrode is then able to create these pulses and are able to be taken into results. The most common type of biopotential electrode is an ECG electrode, which is made out of either silver or silver chloride. They're formed by depositing a thin layer of silver chloride onto a silver electrode, as mentioned earlier, and are used in ECG electrodes, which is used to measure the heart and the heart pulse. An EMG electrode is a circular disc electrode used for the surface. However, they are also able to be used internally with needle electrodes. Needle electrodes can either be unipolar or bipolar, shown in the figures on the left and right at the bottom, either to collect nerve or muscle fiber readings in the form of electrical signals. Another type of electrode are EEG electrodes. These could either be cup electrodes or subdermal electrodes, which are seen on the left and right. They are both used, however, for EEG recordings, but they are just made of different materials. Cup electrodes are filled with electrolyte gel, while subdermal electrodes are platinum or stainless steel needles. They intervary depending on what they are needed to be used for, with the subdermal electrodes used for deep brain interfacing. Lastly, we'll be talking about microelectrodes. They're able to record action potentials on single cells and are extremely accurate. They're commonly used in neurophysiological studies, and the ultrafine tapered tip can be inserted onto individual cells and are meant specifically for that use. However, due to how extremely accurate and small they do end up costing a lot of money for scientists to use.